for the invitation to come. I'm pleased and I'm very eager to learn what is known now about the relationship between milk and immunity. My work was done some years ago, and it has a long-term theme of the evolution of lactation, one could say among mammalian species. Of course, we know there is great diversity in the lactation among mammals. There's great diversity in the composition of milk. There's great diversity in the length of lactation. There's great diversity in the degree to which mothers use maternal reserves. There's great diversity with regard to the size of weaning at the end of lactation, and there are many social aspects that interrelate with lactation. For example, I just sort of put together a table which has categories of milk fat, and you can see that on the far left-hand side there are very low fat milks which include human milk, and on the far right side there are very high fat milks which include some of the seal milks. But the point is that even though there's many species that are clustered down near the lower fat content, there also are many species, not just the seals, that have high fat milk. I did work early on in the 80s on black bears because I was very interested in this species in terms of the offspring are born in a den. The mothers give birth in middle of January. The top photo on the left shows someone putting a pole in with a drug to immobilize the female bear, and that the water and nitrogen cycles from mother to cubs during the lactation period. It's actually a phenomenal system in which the mother is in the den, she's lactating, she's consuming the excreta of the young, but she is not feeding or drinking herself otherwise for months. Somewhere there's a way to do this. There we go. And if we look at the black bear cubs of different ages, growing from newborn to 1 month to 2 months to 3 months, you'll see they change a lot in size, but they're all dependent on milk. And they all actually have very different lactation dependencies based on the mother's activity. The cubs are very poorly developed or altricial at birth. They switch from a placental glucose and amino acids to milk, fat, and protein when they're born. The milk is mostly low in carbohydrate. It's mostly oligosaccharides. We've had a lot of work done on this in the last 20 years. And the milk fat increases. The milk is low in protein. All of these things we could discuss in detail with regard to this species and how this species has evolved a lactation strategy of its own. Low milk yield and reduce the glucose demand on the fasting mother. So each species has different lactation strategies. We worked on California sea lions, which you can see the upper photo is a breeding beach, and you can see all of the animals that have accumulated there. The mothers come ashore and give birth. So we end up with basically a riot of pups on the beach, moms coming ashore trying to find them, and then after nursing going out to sea to feed for several days. So the foraging trips of the mother are important not just because she's acquiring nutrients for herself, but the pups are fasting when she's gone. So they have an intermittent fasting approach, which again is different from what you would think of for many species. And the mothers return to nurse their pups for about 1.5 days at a time. And this continues for 11 months in this species. So the milk intake is intermittent. Fat is moderately high. There's no lactose in the milk, and there's no lactose-based oligosaccharides. There is some reason to relate that to their period of going to sea and maintaining lactation at sea. The milk intake during the nursing periods is about 30% of pup body mass. But again, there's intermittent fast, so the pups are only consuming a small amount on average. And again, what does this do to the microbiome? And again, since the pups are very active and have a high metabolic expenditure, they're very inefficient in growth. In other words, they have to have a lot of milk energy and protein to grow. And as you can see in the left-hand graph, that these are male and female pups, that only about 16%, it doesn't matter, only about 16% deposition. So sea lions and fur seals as a group of animals produce high-fat milk that reduces the milk volume that they have to secrete at sea and enables rapid energy transfer to the pup when they come ashore. Milk secretion itself has been shown to be up and down regulated when the mother's ashore and at sea. And the foraging trips of the mothers in some species are 2 to 3 weeks. Now, what that means is the pups are left on the shore to fast for 2 to 3 weeks. So again, a rather remarkable lactation strategy, the consequences of which we don't fully understand. And it's believed that the loss of alpha-lactalbumin is related to the ability for these females to sustain lactation. Another species that I worked on were hooded seals, which give birth to well-developed pups on unstable pack ice off the coast of, in our case, off the coast of Canada. And the picture to the lower right shows the ice flow and shows our research vessel. Obviously, I mention this because this is one of the species that's considered very susceptible to climate change because they are dependent on having ice that is rafting ice for the mothers to give birth to their pups on, and they have to have it within a very tight time frame. This is a species with a very short and intensive lactation. So unlike the fur seals and sea lions, you might think all seals are similar, but these are the members of the true seal family, and we demonstrated for the first time that the lactation duration is 4 days or less, and that there's frequent nursing and mass gain of the pup of about 7 kilograms per day, which when I first presented this data to some pediatricians, they could not believe. How could an animal absorb that amount of fat in a day? The milk is probably more aptly called heavy cream because you can see that it is 56%, 61%, and 61% at the 3 sort of quote-unquote stages of lactation, which is 0, 2, and 4 days. This picture is one for me to just talk a little bit about. The males have a bladder. They blow out their nose and they shake it up and down to, I guess, scare off other males and to attract females. And it makes sort of a ringing sound as they do this. And this particular photograph shows a female with a dart in her butt because we were in the process of collecting milk from her, and then we had this disturbance. And we, of course, had to stop everything and take pictures. And this shows that they manage extreme efficiency in deposition. The fat transfer from to the pup is deposited at about 88% rate. So it's obviously that and this is a 4-day-old pup on the right side. In the photograph, you can see that he's very obese. In fact, at this age, they can't swim. They can just float like a cork in the water. So it's actually what they do is they're getting onto the ice, and then the ice is carrying them south. So if they didn't have ice, they couldn't swim, they couldn't feed, and it would be devastating for this species. And it's an extreme example of mammary lipid secretion. So I wanted to just show you some examples of very big differences among different species of wild mammals. And I worked on about 75 species, so I know they're very diverse. But the bigger story is how lactation came to be, and that's what the rest of my discussion will be on. Lactation is a defining characteristic of mammals. In fact, the word mammal comes from mammae, and it was from the way in which they realized that lactation was what linked all mammals together. There can be a long dependence and delayed functional maturity of the young, which is important in many different species, whether you're talking about primates where the mothers are holding the infants, or whether you're talking about animals that are using nests. There is a possible transfer of nutrients from one time and place to another time and place, like some of our seals, which are going off at great distance. I didn't mention, but some of these seals are swimming about 1,000 kilometers to go get to foraging sites and then come back to their pups. Obviously, that's only possible if you have some way of storing nutrients and transferring them, which milk is. And how did such a radical approach, especially compared to the pre-existing taxa, evolve? Now, I don't need to elaborate on the fact that there are many novel aspects of lactation. There are novel feeding types, based on a skin secretion. There's the novel gland type. There's novel constituents in milk, which have novel functions. There's novel degree of upregulation of secretion, novel degrees of neonatal immaturity, which are dependent on milk and lactation, and novel digestive features in neonates. How could such complex novelty arise? This was originally raised as a challenge to the theory of natural selection, and Darwin ended up having to put together a chapter in his Origin of Species to try and respond to how it was possible that you could have all of these things having evolved at once. And again, as I'd mentioned before, there's many novel constituents. I'm not going to go into them in great detail, but obviously there are things which are unique to milk that are not found outside of milk and that are genetic products of the mammary gland, including alpha-lactalbumin, beta-lactoglobulin, whey acidic protein in some species, late lactation protein in some species, and so there are some, and the caseins, of course, and some of the milk fat globule membrane proteins that are all indicate a long period of evolution and the evolution of many different constituents. There has been newer evidence, which I will briefly allude to, on molecular evolution, the evolution of specific milk constituents, looking at the evolution of the development of the mammary gland, which again shows some very interesting parallels across taxa, and paleontology, which I will just show you a few pictures rather than go into detail. But obviously, I believe that evolution of lactation has a very big part to play in the paleontological history of the mammals that arose. So a few questions. How and when did mammary secretion originate? Was it before or after the origin of mammals? We don't think of that often. We usually think of lactation and mammals. We don't realize that maybe the two aren't the same. What evolutionary changes have occurred in milk? And what role did lactation play in the evolution of mammals? All, again, are actually talks in and of themselves, and I'm just going to go over them quickly. But first, we need to have an idea of what the time frame we're talking about is. This is a model of the Milky Way, and that line points to the location of our star, of the sun. And this whole thing is rotating about every 220 million years. And so about 3 of these galactic years, before i.e. the 220 million years, were the first multicellular animals. About 2 giga-years was the jawless fishes. About 1.5 giga-years, the first synapsids. These are the ancestors of mammals. And about 1 giga-year, complex lactation evolved. So there's been a long period of evolution. One of the most important initial evolutionary steps was forming a skin that was like the skin that would be important for the evolution of mammary glands. This was had a cornified layer and maturing granular and spinous layers with a proliferative layer. This is a frog skin. The next step is a truly glandular integument, where you have, I guess I can't point, where you have glands enlarged and protruding down into the skin, and in between them there are scales. So this is this is an illustration of a caecilian, which is, there are now known to be caecilians which produce a glandular skin that when the young are born, the young scrape the skin of the mother and get nutrients, fat and nutrients from the mother. Very similar to lactation in some ways, but very dissimilar in other ways. Now this is I wanted to sort of give a bit of an overview of evolution, going back through time. We have time on the left-hand side, so Paleozoic is the oldest time, going up to younger time. And then you have, going across the top, you have different groups that were involved in mammalian evolution, or are relative to mammalian evolution. And basically there was the so-called synapsids who formed the ancestral group for mammals, split in the Carboniferous around 300 million years ago from the sauropsids who produced all the reptiles and birds, what you know of as reptiles and birds. So reptiles and birds are a long, divergent lineage, and not very informative in terms of trying to understand the evolution of lactation. In the middle of this thing, you'll see the little bursts of blue lines, which are just simply radiations of the synapsids. In each case, there was a radiation of the synapsids, and then most of them went extinct, and a few remained. And the same is true of the mammaliaforms at the end. Most of them went extinct, but a few remained and were, in fact, the ancestors of mammals. And the other thing in here in the middle is you'll see there are some schematics of glands with hairs sticking out. I'll come back to these because this is a very interesting phenomenon, is the structure of mammary glands in different groups of mammals. So this is this fundamental divergence that occurred around 300 million years ago, where the sauropsids, who are ancestral to reptiles and birds, separated from synapsids. They're ancestral to mammals, and had very different soft tissue characteristics based on what we understand of the fossil record and the ancestral descendant forms. Here's another example of something that I showed, that, again, going from the left up towards the right are these radiations of different taxa. And again, showing that they existed for certain periods of time, then they went extinct. And it was only the mammaliaforms that actually led to the mammalia around 150 million years ago. And then you'll notice that the whole question of where is the origin of lactation in all of these, among all of these radiations. Now, looking at the size and the estimated egg mass of the mammaliaforms who became extremely small, we have argued that they must have been lactating. If you look at the photograph to the right, you'll notice there is a paperclip. That's a paperclip for scale. So that is the adult size of an adult female, and is of similar size to the smallest mammals that live today. We believe that this was part and parcel of the transition that led to mammalian lactation as we know it today. So these Triassic mammaliaforms were very small, endothermic, estimated from fossil evidence, unable to produce a large enough egg for endothermic young. So therefore, it's been argued they had small eggs with immature offspring. And they also had limited tooth replacement. In other words, they had lost the frequency of tooth replacement so that the young couldn't eat solid foods. And so all of this suggests they must have lactated. And so, when we consider the evolution of lipids, proteins, and sugars, we have to go back a long period of time and not just to the beginning of mammals. Now, molecular evolution, of course, there is evidence of variation among species and suggestions that at a genetic level that different compounds have derived from earlier compounds. And, in particular, I will talk a bit about casein micelles. I will talk a bit about milk fat globule membrane constituents, a bit about alpha-lactalbumin and lactose synthesis, a bit about beta-lactoglobulin, and a bit about whey acidic protein, which is found in rodents and some other taxa. Now, I'm sure you've all seen a model of what the casein looks like, how big it is. And how it is composed of a variety of different types of caseins that form this structure, huge structure, including calcium and phosphorus. A very, relatively speaking, a massive structure, and very important to the transfer of protein from mother to young, and calcium and phosphorus. Now, this is just a graph showing the variety of genes that are in a similarity relationship to casein genes, and studies that have done that suggest the origin of caseins from earlier secretory calcium binding phosphoproteins, and suggestion that they were involved in regulation of mineralization, and could even have been involved in mineralization of eggs. Of course, we all know in this room that the caseins precipitate as curd in the stomach, and that this has an important effect on the stomach environment, and the way in which these things are broken down can have effects on microbial communities, and it involves the gradual release of protein and fat to the intestines, which is an important feature of milk digestion. Of course, lactose, which is in these milks, requires the enzyme lactase, and so really we are dealing with a coevolution of multiple factors of lactase and lactose, of caseins, of proteins, of fats. Everything is interrelated in terms of their evolution. This is just a schematic of a adipocyte, or a secretory cell, mammary secretory cell, and on the right the various proteins that are involved in penetrating the milk fat globule membrane and associating with underlying lipid layer. One of the features of milk fat synthesis is the pinching off of these globules of fat. And one thing that occurred to me was that, of course, if you have a high rate of milk fat synthesis, you can't have a high rate of cytoplasmic loss with the milk fat globules, because that would basically undermine the secretory cell. And so it seems that this was a major aspect of the evolution of the milk fat globule, was the ability to minimize the cytoplasm that is lost with the milk fat globule. And that specific proteins have evolved, and I'm not going to talk about them any length here, but to help hold the membrane tight to the underlying fat layer, and that this is no doubt part and parcel of this mechanism of reducing cytoplasmic loss during milk fat secretion. So when we talk about the evolution of lipid secretion, we have to think about all of the various constituents that go into each of the structures that are part and parcel of milk, including the constituents that are involved in the milk fat globule membrane. So it was a key step in the upregulation of fat secretion was minimizing the amount of membrane and cytoplasmic loss. Of course, I should add here that we don't really have much information on many species. For example, we have these hooded seals that I told you about, had this tremendous amount of fat secretion, and we don't really know how they manage that, how they accomplish that. And that's true of any number of different living taxa, that there are species-specific solutions that have developed. Now, of course, we know that lactose is an important constituent of cow's milk and most eutherian milks. But it's a lot less, it's a lot less major in its import than I realized when I first started working on lactation. Lactose is not found elsewhere in nature, and it requires a special modifier protein, alpha-lactalbumin, that allows GAL-T1 to add galactose to glucose, and that's unique to the mammary gland. Not known elsewhere, to occur elsewhere in nature. But what this slide fails to recognize is that the secretion of lactose may or may not be the final step. It's thought that alpha-lactalbumin is derived from C lysozyme, which is an antimicrobial protein in skin secretions. And it's in fact true that there are multiple constituents that are found in milk that are thought to be related to the earlier immune function earlier in evolution. And again, the red line merely points to the separation of the sauropsids and the synapses as the date which had previously been suggested based on molecular studies. Now, obviously, if you're going to have lactose, you have to have lactase. I mean, it's so silly, right? If people don't have lactase, you better not feed them lactose. Same thing with California sea lions. If you give lactose-containing formula to a California sea lion, you will produce a violent diarrhea very quickly. And that has been known for many years because it was so obvious. Obviously, we need to have both lactase and a lactose. If we're going to digest lactose, we need to have lactase, but that would mean that we need to have evolution of lactase if we're going to have the evolution of lactose. And again, so this is the interactions of different constituents that are important because of their interrelationships. And then I guess the question was, how did lactose evolve in milk if there was no lactase in the neonate? And vice versa. It's the chicken and the egg. You know, you have the lactose but no lactase, you got a problem. and vice versa. If you have lactase and no lactose, it doesn't do you much good. So among mammals, we do know that there are species that do not have lactase and probably cannot digest free lactose. Many of them, however, have fairly high levels of oligosaccharides in their milk. And that has perhaps been, over my career, the biggest change we have seen is this recognition of the tremendous diversity, not just in human. Of course, human milk has a greater diversity of oligosaccharides than any other mammalian milk, as far as we know. And we've looked at other primates as well. But there's a lot more oligosaccharides than people recognize. In my earlier slide, I mentioned about bears having oligosaccharides in their milk. And they have long, complex structures. We don't really know the function of these structures. We do not know, and I asked this group to come up with answers. We don't know, with fucosyl oligosaccharides or sialyl oligosaccharides, what effect they're having, how they're being digested. I thought many years ago that, well, the oligosaccharides were being taken up into the enterocytes and broken down there, but it's now hard to know because we know there's such a great diversity of oligosaccharides. So here we have basically a big unknown about mammalian lactation. And there's a lot of papers coming out, but what the significance of them biologically or in terms of microbiome is not certain to me. And the sea lions and fur seals have secondarily lost the ability to synthesize lactose and cannot tolerate lactose in formula. Now, we've recently been trying to look at some of the quote-unquote ancestral forms of mammals, in particular echidnas, which are an egg-laying species. Okay.
That's better.
And have come to look at lactose as a substrate to which other constituents are added. Because we see this, in fact, that there might be a small amount of lactose and then a large number of compounds that have been added, and that the enzymes that were involved in forming these oligosaccharides were have already existing and had many functions. I was particularly interested in the whole idea of whether milk it's been something I've talked about for some years, whether milk could have evolved in relationship to eggs and providing a surface secretion to eggs. And we do have compounds in echidna milk which are antimicrobial in nature. So they are obviously doing something, and in fact the predominant sugars are 4-O-acetyl-3'-silyl lactose, which restricts microbial metabolism. Again, it gets very complicated when you think about the fact that the echidna gives birth and then keeps the egg in a pouch, and then they're lactating or producing secretions which are being absorbed by the eggs, because these eggs, actually echidna eggs, before they are laid, are a parchment shell type that swells with fluid. And so, the question then became is, could it be that this was an original function of oligosaccharides that secondarily, later on, became providing lactose as a source of nutrients to, particularly our group, to eutherians. And again, but that's just hypothetical. This is just a list of milks that are high in oligosaccharides and little, if any, lactose. The echidna and the platypus are, the primitive egg-laying mammals. All known marsupials, those are kangaroos and their allies, have milks that are high in oligosaccharides. All bears and true seals have milks that are high in oligosaccharides. Many carnivores, such as skunks and mink and coatis and raccoons and cats, produce milk that are high in oligosaccharides. Now, I should point out that these are not all the same oligosaccharides, so that the oligosaccharide pattern in a striped skunk will be different from that in mink. So we have this variation among species in the pattern of oligosaccharides. without really knowing what the significance is. Many primates have both lactose and oligosaccharides, and it's not clear why there is such diversity in oligosaccharides among mammalian milks. It's a slow and tedious process to add species to the list. Now, other proteins are very interesting from the point of view of evolution. Beta-lactoglobulin is of a class of proteins known as a lipocalin, which have a defined structure, and these have been looked at in terms of their relationships to each other and other lipocalins. And all you need to get from that is that the beta-lactoglobulins were among the earliest diverging of the lipocalins, and they apparently diverged earlier than other lipocalins that are found in fish and things that don't make sense. But the point is that each constituent in milk has potential significance in terms of trying to understand its evolution and biological importance. And whey acidic protein, again, is another protein that is found in rodent and other milks. The available evidence is that it was a quote-unquote ancestral protein that's just been lost in many mammals. And that seems to be true of other milk constituents. It is a ancient origin, this 4 disulfide core domains that define this group of proteins, and they again seem to be related to antimicrobial and protease inhibitors, and so they're very interesting and probably overlooked because they aren't found in either dairy animals or humans, lactation. But that doesn't mean they might not be a very valuable constituent to look at. I don't know about guinea pigs or things like that, but. Lastly, I want to talk a little bit about one of my pet stories, which is about the evolution of glands in mammals. Of course, this schematic on the right is showing the hair follicle, and then associated with the hair follicle, a sebaceous gland and an apocrine gland, and not associated with the hair follicle, a eccrine sweat gland. But from a developmental perspective, dermatologic developmental perspective, the hair follicle, sebaceous gland, and apocrine gland form a developmental unit, the APSU, which when I first discovered, I didn't quite understand the significance of this. But I thought it was, very interesting when I'm trying to think of what did mammary glands evolve from, and in my earliest days, I thought they were derived from apocrine type glands. And there's no doubt that mammary glands are one gland. It's one thing you had to worry about, of course, is could mammary glands in different taxa be different, structures, something that evolved separately from different origins. But based on histology, ultrastructure, secretory mechanisms, biochemical pathways, the secretory products, milk, and the importance of oxytocin for milk ejection, it is clear that they're all the same type of mammary gland. So the mammary gland goes back to a common origin. Looking at some of the weirder mammary glands, this is a echidna mammary gland, which is, the gland has been dissected out, but it consists of 100 to 200 lobules, each of which drains separately, and each of which empties into the infundibulum of a hair follicle. So there's mammary hairs sticking out associated with each lobule, and these mammary hairs are bigger than the hairs in the surrounding pelage. So they are distinctive. I guess the only picture to focus on is the middle one here, which is the egg. And you can see the egg is being held between someone's fingers, if you see the fingernail there. So that gives you an idea of how large these are at hatching and how small the eggs are. It is actually quite interesting that in the earliest phases of mammary development in the echidna, the mammary gland looks very much like a skin gland. And then it develops and proliferates into that either substantial structure. And this is just a cross-section of a gland that shows that there are mammary hairs on the right-hand side. Mammary hairs and mammary hair follicles embedded in the gland itself, that they're not outside the gland, they're part and parcel of the structure of the gland. There's a developmental association with a hair follicle and a sebaceous gland and a mammary gland, suggesting that the mammary gland is related in terms of the developmental position to an apical gland. This is a turn of the century, last century, drawing of the development of the mammary gland, and I don't really want to go. It's starting upper left is the embryo, and then the gland spreads out as a plate, and then it develops little projections that come down, and each of these projections that comes down becomes one of these mammary structures. So it's in marsupials, which are like kangaroos and their allies, and in the middle there's a picture of a so-called joey, in other words, a baby kangaroo attached to a nipple. They actually do attach to the nipple, and for a period of time, after they've attached, you can't get them off. Or if you get them off, you can't get them back on. So that's interesting because it changes the dynamics of access of microbes, presumably. So the kangaroo is live born, but they hatch from a eggshell membrane in utero. So it's sort of like there's an egg-laying process, but they hatch before. There's minimal placental structure and transfer, so it's mostly post-partum, it's mostly mammary gland. There's a variation among different species of marsupials in the birth size and state and placental function. But the milk is, where it's been studied, is always dilute, containing lactose and monosaccharides. I should point out, as the young get older, the maternal mammary gland converts to producing oligosaccharides. So it's a change in function. The milk is no longer a lactose and monosaccharide, but an oligosaccharide-rich fluid. And there's low levels of casein and whey proteins and little lipid in the initial secretion. I will try to explain this. This is on the left-hand side of each of A, B, C, and D is a little diagram. And the right-hand side is something that's actually from a histological, drawing from based on histological section. The mammary gland starts as a sprout that elongates downward. If you go from A to the one to the left side of B, and then there are mammary glands that sprout out of, and hair follicles that sprout out of the same developmental structure. And then in C, the nipple hollows out, and the hair follicle that's associated with the mammary gland is actually projecting into this hollowed out structure. And then the nipple everts, and the hairs are shed, and now the galactophores transfer the milk, were originally where the hair follicles had been associated. So it's a very unusual thing. In fact, you probably can't see it, but on the lower right-hand side is from a koala, and there's actually a hair after the nipple is everted. There's a hair sticking out, which I always thought was very exciting because it's not something we can imagine that this is the neonate is, producing nipples with hair follicles sticking out of them, and then they're shed, and then later on there's mammary development. And this was one of the things while I was working on a review of this that I was especially surprised by, was the recognition that if you look at the human mammary gland development, and this is not found in most of the developmental biology texts on mammary glands, there are also sprouts and hair anlaga developing from the developing mammary gland, and so that in the lower left-hand side you can see these things sticking out. They're actually clusters of what will become secretory tissue along with sebaceous glands, along with hair anlaga. And of course, these things are shed later on, but to me it shows the relationship that appears to be in terms of the evolution of mammary glands across taxa. And this is just a schematic of some of these things, which just sort of shows on the top mammalian pelage, which has the APSUs, hair follicles, and apocrine glands, and then the developmental stages in 7 species, showing the MPSUs, the mammary gland pilosebaceous unit, developing at various stages of development in these taxa. So again, we have not recognized the middle lower left is human, and that hasn't been recognized as an important stage. And of course, in mouse and cow, you don't have these stages, so they've been ignored. So anyway, with that, I want to thank you for your attention. These are Waddell seals, and I put on the bottom the world's southernmost site for lactation research, because I was there.
Thank you so much. We have about 10 minutes for discussion and questions. Dr. Mistiki.
Thank you for interesting lecture concerning lactation. But as we talked last night during a dinner, You know, production of external secretions, which are of nutritional and immunological impact, are not restricted only to mammals. Those of you who are aquarists probably know that the crown jewel of all aquariums, freshwater, is a fish called Symphysedon discus, beautiful Amazonian fish that big. And it produces mucus on its surface like all other fishes. But in this case, it's very unique because when the little fish hatch, they attach to the skin of the mother and father for a few days, and they eat the product that is of lactating or of glands in the skin and survive. If they don't do that, they die. Yeah. So it's passive, I guess, immunity. And so why I say immunity? This fish was very expensive because in captivity they didn't propagate that easily. But some smart guy took regular chicken eggs, mixed egg yolks and egg whites, put it on the bucket, let it dry, filled it with water, and then added the fry of the newly hatched fish. And they survived. Due to the fact, as you probably know, chicken eggs contain lots of antibodies. Actually, egg yolk is full of IgG-like molecules. So that's resulted in a tremendous decrease in the cost of these fish. And now we have varieties which do not even exist in nature. The second species, birds, mammary gland of the birds, that is the crop, which is histologically very much similar to the gut. The birds collect the food, store it in the crop, and then regurgitate it to the newly hatched birds. But it's covered with antibodies because the gland of the crop is full of antibody-producing cells. They are secreted into the lumen of the crop and therefore provide passive immunity. And actually pigeon milk, which is a term known in ornithology, is very much similar in its constitution to the milk.
Thank you very much. We actually did some work on, I had a student who did a master's thesis on pigeons, and so it's very fascinating, but I love your perspective on it.
Well, we're at it. You gave an elegant discussion of the anatomic evolution and the chemical composition of the different species of milk. We didn't talk about immunoglobulins or cellular content of any of these. Do you have any thoughts about any of the distribution and patterns of evolution of immunoglobulins? Some of the animals don't have IgA at all. The sea animals have some IgM. They have nothing else. They haven't grown up. Have you have any feeling about the evolution of immunoglobulin and cellular distribution of either antibody or T cell function in other species?
My only observation is I wish I had started on that back when I started on lactation. Because I think there's so many important and interesting questions, and I think that,
Yes.
I think it's reasonably well known. The first antibody in secretions, even starting with the fish, is IgM-like molecule, not IgA. IgA is a latecomer only in birds. It starts in birds. So it's IgM and then IgG-like molecules, like skin of the shark, secretions over there. It's IgM and IgG, but not IgA yet. So there is a...
There was a question in the back. Oh, Dr. Walker.
You mentioned, as you were going through the evolution, microbes. Is there a sequence of events where microbes are more involved in lactation and the content of lactation as we go through the mammalian species?
Yeah, I think that...
Closer to the microphone, please. Thank you. Closer. Yeah, closer.
I think that it has been largely overlooked. I think this is something that's very crucial to the evolution of lactation, and I think that, may have been behind the evolution of carbohydrates in milk and many other constituents in milk, which were what became obvious to me in examining the evolution of constituents in milk, as many of them are antimicrobial originally. Maybe not now, but I think historically or evolutionarily, that this is a very important relationship, and I wish that we understood it better across taxa, because I think we have this natural experiment, which is all of the different varieties of mammals that produce milk, and that of course have to deal with microbial species in the gut and elsewhere, and whether there's been, how much of this, was driven, how much of the evolution of milk constituents was driven by a need for defense, and secondarily became more important for nutrition.
There was a question in the back somewhere. No? Well, thank you so much. It was a remarkable presentation, and we really enjoyed it very much. Thank you.